Yeah. First, welcome here. So I'm asked to introduce one of the experiments about the protein related. Before that, I'd like to introduce the team. Laura from the Oakland. Laura, thanks for coming here to help with the workshop. And also Roisin is a PhD student working with Norm and me. And she's helping this session too. As introduced, I'm a science instrument scientist at NCR, but I have a research interest on the protein dynamics. But before I get into the details, what we are going to do for the workshop for the protein part, how many of you have heard of proteins? Great. I think I will have more hands than polymer people. Okay, anyway, that's a joke. And so because today, and we are only focus on certain things about the protein dynamics, we try to study the neutron spin echo. I thought that to give you a flavor that there are many other things that neutron spin echo can use to study protein system. So people have been using the basic neutron spin echo to examine some liquid series, study concentrated protein solutions. And also, people have been using the neutron spin echo, look at the so-called reversible cluster formation in solution. You may be wondering why this is interesting, because actually that's related to the pharmaceutical problem, exactly understand what's the mechanical mechanism that are related to the viscosity effect that affect many of the cancer treatment drugs. And also people study some fundamental physics related to this. For example, when protein goes through some glass transitions, there are local dynamics. They can study that with neutron spin echo. But today and in this workshop, we'll use example to study the protein internal motions. Okay, so why it's important to study internal motions? And it's because that many proteins are very flexible. You don't need to know what exactly this protein is, but this basically people take a cryo-EM imaging and look at the same protein at a different condition. You can say their shape is different. So why is this flexibility important for the protein functions? So I'm not a biologist, but I happen to come to this paper. I just published it last year. So they studied how one type of protein called monoclonal antibody, how they bind to this COVID virus and say that why that particular protein is important. So they end the conclusion that actually they have different way of binding to the COVID virus. And they reach a conclusion that they binding through this called conformational dynamics. But the problem for that is that they can only say structure. When you talk about dynamics, you really have the motions, but how do you directly probe that? That's where the question, and we hope to use neutron spin echo to address. But when looking at the protein internal motions, actually it's very complicated. Okay, so because the protein structure is complicated, we have chemical bond. People could look at the bond vibration. And from primary structure, then you have some molecular motions, side segment motions. And then once you from the secondary tertiary structure, they start to have the domain motions. For different type of motion, because the dynamics and non-scale are different, the technical use are different. So today we are focusing on is this large domain motion. Because the large domain motion actually is quite challenging. When we say large domain motion, what do we mean? So it means that usually they form the tertiary structure already. The size of the domain is about a nanometer or more. And then because the size is relatively big now, the relaxation time and the time scale, we're talking about a few nanoseconds to tens of nanoseconds. And actually looking at the motions at this length scale and the time scale in combination, it's very challenging. So as far as I could know, there are no other technique. And the only answer is, I guess you know the answer, right? What's the answer for that? What instrument should you use to look at this? This should be 100% correct, right? The answer is neutron spin echo. So of course, the information I listed here for the previous version of neutron spin echo, before it's upgraded, so we can reach about 300 nanoseconds. And length scale-wise, from 3 angstrom to tens of nanometers, that's perfectly fit to this type of applications. So example we choose for this workshop is called a monoclonal antibody. Anybody heard monoclonal antibody? Some of you raise your hand. I see how many of you heard. Okay, there's a few. Good. And so basically, to me as a physicist, I do not know the details. To me, it's just Y-shaped particles. They have 3 domains, okay? So they have one FC domain, and then they have 2 FAB domains. 2 FAB domains are identical to each other. Of course, now industry are getting better and better. They can make it, actually the 2 FAB domains different, the bispecific proteins. And so why this type of protein are important? Actually, if you look at basically the business for the different companies for their bestseller of the drugs, many of them are based on monoclonal antibody. One of them showing that 2017, there's an annual sale is about $80 billion. Obviously, you can say it's a very important molecule, but however, our understanding is still very limited, I have to say. 2 minutes? Okay, thank you. And well, there are many monoclonal antibody, and particularly for this spherical workshop, we'll use the one so-called NIST map. It's a standard monoclonal antibody that basically actually proposed by NIST and used by both industry and academic research. And many companies and actually study these molecules as standard molecules. And so I just want to show you this simulation results. And this all atomic simulation. Of course, I only show the protein without a water molecule is how it's moved. So, but that's translational motion. But what we are interested to measure, if a fixed center mass, and then you consider the domain really not rigid, how do we measure quantitatively this domain motion? That is the question we try to address from the work today. So I'm not going to details all of them. You will see the data later today, and then we try to, from that, to tease out the information quantitatively, what's the domain, internal domain motion for this molecule. Just to let you know that there are many other studies of the internal motions of protein, and so I'm not going through them. And there are some review articles here. And with that, I hope to engage you more conversation with you. If you have any questions, even you are not in the protein group, feel free to ask me or anyone in the team, and we'll be here through the workshop. Thank you.